Let's continue on with cellular level of organization, looking at nucleic acids and protein synthesis. We need some definitions here. So, terminology: genetic information instructs the cell on how to construct proteins. That genetic information is stored in DNA. Your DNA is found on the chromosomes in the nucleus of each one of your cells. A gene is just a segment of the DNA that codes for one protein. A genome is the complete set of genes, all the genes that make up an individual. The Human Genome Project took many years, many scientists, and big computers to map out all the genes in a human being, and that was called the Genome Project. Complete set of genes. Genetic code is a method used to translate a sequence of nucleotides of DNA into a sequence of amino acids, and the sequence of amino acid would result in a specific protein. And that is called the central dogma of genetics. Now, what happens in the nucleus of the that genetic code is read, and as it is read is called transcription. A complementary RNA molecule is formed aside the unzipped DNA molecule, and that's called transcription. It's complementary to what the DNA is telling it to do, and that messenger RNA leaves the nucleus and goes out into the cytoplasm, attaches to a ribosome, and then it is transcribed, being read into a sequence of amino acids, and when all amino acids have been delivered, then it results in a protein. We'll talk more about that. Okay, so that's the central dogma of genetics, is DNA to RNA to a protein. An individual receives half of their genetic information from their mother and half from their father. And let's say that you have blue eyes. Your mother has blue eyes, but your father has brown eyes. Let's talk about that for a minute. Now, your mother has blue eyes, and that is a recessive trait. So she received from her parents a gene for blue eyes from both parents because it is recessive. Now, your father is brown-eyed and you are blue-eyed. So that means that he received the brown-eye trait from one of his parents and the blue-eyed trait from another parent. Now, why can we say that? Well, you have blue eyes, and that's a recessive trait, so you have to have two of those genes to have the blue eyes. But he has brown eyes, so that means we know his genotype. His genotype is said to be hybrid for that trait, a dominant and a recessive. Brown eyes is dominant over blue. He's carrying the blue-eye trait, but it's masked by the dominant trait of brown eyes. That is his genotype. Your genotype would be two recessive traits for blue eyes. Your mother's genotype would be two recessive traits for blue eyes. Now, the phenotype. Now, this term means the appearance of that trait. Now, your dad has brown eyes, but his genotype is hybrid because you have blue eyes. Therefore, we know that he has the brown eye trait because that's what's being expressed. That's his phenotype. His genotype is hybrid because he has a brown eye trait and the blue eye trait together. So often the phenotype shows what is dominant, or in your case, what is recessive. We know your genotype by your phenotype, but it's hard to predict with your dad if he would have had two dominant brown eyes, one from mom, brown eyes, dad, brown eyes, then you would have brown eyes, and you would be hybrid because your mom would contribute a blue-eyed trait. Okay? So genotype is the genetic makeup and the phenotype is the appearance. Now, the building block, the molecule structure of DNA, the DNA is a double helix. This is called a double helix, okay? It has this spiral shape, and it has two halves, two complementary strands. The strands are bonded together via nitrogenous bases using hydrogen bonds. And here we have hydrogen bonds, and these dots here represents hydrogen bonds because hydrogen bonds are easily broken and easily built. Okay, so it is a double strand of two complementary strands of nucleotides. So the building block for a DNA is a nucleotide. You have a sequence of nucleotide bases on the DNA that determines the sequence of amino acids in a protein. And those nitrogenous bases, because they are nitrogen containing, nitrogen, so they're called nitrogenous bases. Adenine, thymine, guanine, cytosine are the four nitrogenous bases found on a DNA molecule. Now, the adenine and the guanine are called purines, and the thymine and the cytosine are called pyrimidines. You don't have to remember these terms, but it may help you to know that A, because it is a purine, will only match up to a pyrimidine, the pyrimidine thymine. So A always attaches to T, and the guanine, the purine, always matches up with cytosine, the pyrimidine. Okay, so G always bonds with C. Guanine to cytosine, adenine to thymine. A single organic ring structure, so only A and T match up together and C and G match up together. A good way to remember this, because you were at Grayson College, A-T, at Grayson College. So A to a T and G to a C, if that helps you to remember that the AT and GC. There are two polynucleotide chains. There are hydrogen bonds holding the nitrogenous bases together. The bases are paired specifically, the adenine to the thymine and the cytosine to the guanine, and it forms a helix. Now this would be like, alright, so you have a chain ladder, and here are your metal rungs, and you take the bottom of the ladder and twist it, and that would be the shape of a helix. DNA is wrapped about histones and proteins that form chromosomes. Okay, this is the DNA macrostructure, and you can see here's our chromosome, the chromatin, and we can look at the histones, and this is wrapped all around it. Here's that DNA double helix, and look at these matching up. Now we're unzipping that DNA and exposing those nitrogenous bases. So we had A was bonded to the T with a hydrogen bond, the T to A, the G to C, A to T, C to G, G to C, and so forth and so on. So this is how these nitrogenous bases bond together. The strands of DNA are wrapped around supporting histones. In the nucleus, the DNA double helix is packed by special proteins called histones to form the complex called chromatin or chromatin, and these are decondensed. The chromatin undergoes further condensing to form a chromosome. So when it's all stretched out in the nucleus and not normally visible with our compound microscope, it's called chromatin. And if the cell is not undergoing mitosis, then we don't see the chromatin. It's invisible to us. But as it's getting ready to divide, that chromatin will condense. It will thicken up and it forms chromosomes. And we can see the chromosomes. DNA replication, to make a copy of itself. And when does this happen? We've talked about mitosis. When does this happen when you have DNA replication? Well, it's right before the cell begins to move into mitosis for cell division. You have to have DNA replication. So you have 2 sets of chromosomes, one for each new daughter cell. Okay, so what happens is we have the enzyme helicase and DNA polymerase. Helicase will break the hydrogen bonds between the nitrogenous bases. It unzips that DNA, and the leading strand and the lagging strand, you have these nitrogenous bases come in to join up with the complementary nitrogenous base that's already there. And a half of that helix is that double strand, and one or half of that new DNA molecule, one strand is from the original and one strand is newly constructed. And because one's newly synthesized and one is pre-existing strand, we call that semi-conservative. So what semi-conservative means is half is from the old and half is a new part. The helicase here is the enzyme that helps to unzip the DNA molecule.
Stranded DNA molecule is used to create 2 double-stranded molecules with identical DNA sequences. For this process to occur, the 2 DNA strands are separated, and the DNA replication machinery is assembled at the replication fork. The 2 DNA strands are used as template strands to create newly synthesized DNA strands. The key feature that allows a DNA molecule to produce 2 DNA molecules with the same DNA sequence is the ATGC rule, also known as Chargaff's rule. According to this rule, adenine in one strand only hydrogen bonds with thymine, and guanine only hydrogen bonds with cytosine. For example, an incoming nucleotide with a cytosine base will only hydrogen bond with a guanine base in the template strand. By this mechanism, one double-stranded DNA molecule provides the information to create 2 double-stranded molecules with identical base sequences. Each copy contains one newly synthesized strand and one original strand. This mechanism is called semiconservative DNA replication, because half of the DNA molecule is conserved from the original molecule.
Okay, this slide is showing us the central dogma again. Here we have DNA, that double-stranded template strand of genetic information. And through transcription, we can build an RNA molecule. In the nucleus, this DNA unzips, a complementary strand of RNA is built within the nucleus. It leaves the nucleus and goes out and attaches to a ribosome. And translation happens, and amino acids are brought in according to this sequence, all coded from the DNA onto the RNA. According to this sequence, these amino acids are brought in order and eventually to form a new protein according to the genetic code. The DNA holds all the genetic information necessary to build a cell's proteins. The nucleotide sequence of the gene is ultimately translated into amino acids in a certain sequence of the gene's corresponding protein. So how does this happen? Let's talk about RNA molecules. An RNA molecule is a single strand. Remember, the DNA is a double nucleotide strand. An RNA molecule is a single strand. Now, this double nucleotide strand has unzipped, and when it unzipped, those hydrogen bonds were broken between the DNA strands, and it exposed one strand out of the double-stranded DNA molecule, and complementary to it, an RNA strand will be built. Okay, so RNA is a single polynucleotide chain. The DNA has unzipped, and it has the instructions that can leave the nucleus, so the messenger is needed. Okay, so messenger RNA is built complementary to the DNA. On the chromosomes, the genetic information in the DNA never leaves the nucleus. The DNA double strand unzips, and a messenger RNA is built complementary to the DNA instructions. Okay, so here's what's unique about DNA. Our DNA has adenine, thymine, guanine, and cytosine. RNA has no thymine, it only has uracil. Okay, so A, which usually matches up with T, we have a uracil comes in, and hydrogen complementary bond to this temporarily adenine. With this thymine and adenine, guanine, cytosine, cytosine to guanine, guanine to cytosine. It delivers the genetic information from the nucleus to the cytoplasm. So this is called messenger RNA. Once this strand has been fully built and it has the map, it has the code for building a protein, it will leave the nucleus and go out into the cytoplasm as a single polynucleotide chain that's formed complementary to a single strand of DNA. The nucleotides are complementary to the DNA nucleotides. No thymine, you have uracil, and this process is called transcription. The DNA cannot leave the nucleus, but the protein is built in the cytoplasm. So messenger RNA can leave the nucleus and go to the cytoplasm attached to a ribosome so a protein can be built. Transfer RNA carries amino acids to the messenger RNA. It carries the anticodon to the messenger RNA. So messenger RNA has a codon, and the transfer RNA has the anticodon. It translates the codon of the messenger RNA into an amino acid. And then you'll have ribosomal RNA. rRNA provides structure and enzyme activity for ribosomes for building that new protein. So from DNA to protein, so within the nucleus, this DNA is going to unzip, and a complementary to the DNA with its code, the messenger RNA is made in the nucleus, and when it is complementary to the bases, it's called transcription. It's transcribing the information that the DNA contains. It's got that information on this strand of messenger RNA. It leaves the nucleus, attaches to a ribosome, and then a sequence of 3 nitrogenous bases at a time, and those 3 nitrogenous bases are called a codon, is read, and then translation happens when it's read, and that transfer RNA brings in an amino acid according to the codon on that messenger RNA. Transcription through translation. Transcription within the cell nucleus produces the messenger RNA molecule, which is modified and then sent to the cytoplasm for translation. The transcript is decoded into a protein with the help of ribosomes and transfer RNA molecules. Okay, let's continue to review. Transcription is happening in the nucleus where you have a DNA molecule unzips. Okay, this is the original DNA molecule. A complementary strand is built complementary to the DNA, and it's RNA transcript. This is messenger RNA being built complementary to the DNA strand that is unzipped with the help of RNA polymerase. Now, an ASE ending indicates that's an enzyme. So it's an RNA enzyme that helps to move this growing strand along here. So more nitrogenous bases are being plugged in here to this RNA that is building messenger RNA inside the nucleus. So we had adenine to uracil, thymine to adenine, guanine to cytosine, adenine to uracil, and so forth and so on. This is called transcription from DNA to messenger RNA. In the first of the 2 stages of making a protein from DNA, a gene on the DNA molecule is transcribed into complementary messenger RNA molecule. This happens in the nucleus of the cell, and then that messenger RNA has to leave the nucleus and go out to the cytoplasm through the nuclear envelope pores out to the cytoplasm. It will attach to a ribosome, and then translation starts. Now you will see these nitrogenous bases. For every 3 nitrogenous bases, that is called a codon. It codes for the amino acid. Every 3 nitrogenous bases codes for an amino acid. Okay, it's called a codon. So let's look at a codon chart. Okay, these represents those 3 nitrogenous bases are codons. This is a codon chart. Okay, uracil, uracil, uracil for a phenylalanine, and for short PHE, leucine, UUA, and way over here, how about glutamine, is CAA, cytosine, adenine. That is a codon. That's 3 nitrogenous bases in a row on that messenger RNA, codes for the codon. There are 22 different types of amino acids, and 4 types of bases can form 64 different messenger RNA codons. 1 codon is for starting, so we have a start codon here, and we have a stop codon here, and these are also stop. This is an amino acid, but these are just plain no amino acids associated with these. 1 codon is for starting and 3 for stopping, and 2 of those are not amino acids. Nucleotide bases are read in 3s. The 3 nucleotide bases code for 1 amino acid, and that's called a codon. So the messenger RNA carries the codons. There are also start and stop codons. We just said that. And then transfer RNA carries 3 matching nitrogenous bases that are complementary to these that are called the anticodon. Now, let's just take this one right here. And there's CAU. So the anticodon, this is a codon. The anticodon would be what's complementary to C, G. What's complementary to A, U. What's complementary to U, A. So the anticodon would be GUA, would be the anticodon. Okay, this is a chart of the amino acids with the codons. 20 amino acids. Now, essential amino acids are amino acids that the body cannot synthesize, or if it does, it doesn't make enough of them, so you have to get them from the food you eat. There are essential amino acids and essential proteins, and these are proteins that your body's not able to produce by itself. And essential amino acids, your bodies can't make these, so you have to get it in your diet. Some of these essential amino acids are just needed by children, and then some for children and adults. And a person that is trying to follow a vegan or vegetarian diet needs to know how to combine foods so that they can get complete proteins versus incomplete proteins. If they just ate corn, they would get these amino acids and these, but you have to combine them with beans and other legumes to get all of the amino acids needed, those essential amino acids needed to complete the protein. And so the essential proteins are those that you can't get, your body doesn't make them unless you take in these essential amino acids. And so a vegan or vegetarian should be educated, and a lot of times this is if a person grew up in a family that is vegan or vegetarian, then they learn how to cook and combine foods from their parents. If a person is newly vegan or vegetarian, they need to be educated in how to combine foods like wheat bread. You have wheat bread, and that has a few amino acids, but not a whole lot. And then peanut butter has a little bit of what you need, but not completely. You put the peanut butter on the wheat toast, and you have a complete protein. You have to know how to combine foods to get those essential proteins that you need. Okay, translation from RNA to protein. So here you have that messenger RNA, and there you have that 3 nitrogenous bases. That 3 nitrogenous base section there is called a codon. Transfer RNA, and this is a transfer RNA, tRNA here. They carry in the amino acids. By the way, that messenger RNA has attached to a ribosomes 2 units of this ribosome that is helping to move this messenger RNA along, and as it's moving along, then the transfer RNA is bringing in the anticodon to match up. The purpose of the anticodon is like it reads the code here of the codon, so if this anticodon doesn't match, it does have a amino acid that is specific to this particular transfer RNA molecule, and if it's not the right amino acid, it will not match up with the codon. The anticodon and the codon will not match up, but you see AUG, UAC is complementary to this codon. So that's the purpose of the anticodon. So it'll come hook up so that this amino acid can be brought in. And then as the next amino acid is brought in to match up with this codon, then those two amino acids will form a peptide bond. And then so dipeptide to polypeptide chain. We've talked about peptide bonds before and in chapter 2. And then you had dipeptide, polypeptide, and then all the way out to the stop on that messenger RNA till you'll have a complete protein. During translation, the messenger RNA transcript is read by functional complex consisting of a ribosome and transfer RNA molecules. The transfer RNA molecule will bring in the appropriate amino acid in a sequence to the growing polypeptide chain by matching the anticodon to the codon on the messenger RNA.
Translation is initiated by formation of the initiation complex consisting of the smaller ribosomal subunit, the first amino acid tRNA, and messenger RNA. The larger ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl or P site, and the other is called the acceptor or A site. The initiating transfer RNA carrying its amino acid binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The amino acid carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site. The ribosome now advances a distance of one codon, and the first transfer RNA is released. A transfer RNA carrying the next amino acid now moves into the A site, and the ribosome shifts down by a distance of one codon. This is followed by the two amino acids on the transfer RNA in the P site being transferred to the third amino acid. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when the ribosome reaches a codon that does not code for an amino acid, called a stop codon. The ribosome dissociates into the smaller and larger subunits, and the messenger RNA and protein are released.
Now, this can be happening in several different locations on a single messenger RNA strand. So you can have more than one protein or polypeptide chain forming in different locations on the same messenger RNA strand. So several molecules of proteins can be built with a single messenger RNA strand. Transcription factors regulate gene expression. So transcription factors, while each body cell contains the organism's entire genome, we've talked about that before, different cells regulate gene expression with the use of various transcription factors. Transcription factors are proteins that affect the binding of RNA polymerase to a particular gene on the DNA molecule. Mutations. Mutations change the genetic information. Results when extra bases are added. So if a codon's a sequence of 3 of these bases and you stick in an extra base, then it's going to throw all of them off. The bases are changed, and it could code for a different protein than what's needed, or it may not be able to code for a protein at all. may or may not change the protein, because as we looked at that codon chart, there were sometimes up to 4 different codons that coded for a particular protein. So a single mutation or 2 may not change the protein significantly or change the code significantly. So it may or may not change the protein. We have repair enzymes that correct mutations, and if they are functioning, then it could correct the mutation in the sequence of the nitrogenous bases. So if it codes for, originally it codes for glutamic acid here with TTC, and you stick in an adenine instead of a thymine, then that would be a mutation, and it's coding for a different amino acid. PKU, you need to know about PKU for the exam. It's an enzyme that breaks down an amino acid. Phenylalanine is missing, and phenylalanine, especially in a baby, a growing brain, causes mental retardation. It's treated by diets very low in phenylalanine. Phenylketonuria, PKU, results from an inability to metabolize the amino acid phenylalanine. Now, this is one of those tests that newborns are given. There are several different tests that newborns are given, and hospitals will administer this test so that the parent will know right away that there's an issue, and a diet low in phenylalanine could prevent the mental retardation.